Yes, ma'am.
Okay. Now today we will discuss about the gene mapping method. So what is gene mapping? What are the different methods of gene mapping techniques? One second. Okay, students. Now coming to gene mapping methods, you all know actually gene mapping is nothing but it is also known as the genome mapping, and this is one of the method actually which is used to identify the new genes and also to locate the location of a gene on a chromosome and also to know the distances between the genes on a chromosome, not only within the gene but also beyond the gene level. So the gene mapping actually is helpful to identify the gene which is responsible for a given phenotype or to know which chromosome contains the genes and where the gene actually is located on the chromosome and also to understand the function of a gene. So it actually it is nothing but in one way it helps you to locate the genes or to locate the position of the gene, to locate the function of the gene, and also to know which chromosome contains which gene and it is responsible for what function. That is what we call it as nothing but the gene mapping. Now the gene mapping, there are different types of gene mapping methods are there. Okay, so it describes any method used to identify the new genes, the locus of the gene, and also to find out what is the distance between the genes on the chromosome, either in the linkage units or in the physical units. So actually earlier in those days, the scientists, what they used to do is they used to map the genes using linkage analysis. Okay. One second. Okay, sorry for the disturbance, students. Okay. Now, sorry for the disturbance. So now we will discuss on the gene mapping. So you all know, just now I have already told you. So gene mapping, also known as the genome mapping. So actually it will help you to find out the specific point, that is the locus of a gene, and it will also help you to find out the distance between the genes, not only between the genes on the chromosome, but also between different sites within the gene, either in the linkage unit or in the physical unit. Now, actually, these genes, you all know, actually, they are designated to a specific position on the chromosome. That is what we call it as the locus. So, where are the genes located? Actually, the genes are located at a specific point on the chromosome, which we call it as nothing but the locus. So, the closer the two genes are to each other on the chromosome, then the more likely they will be inherited together. So, actually, in those days, they used two different types of techniques or the methods to identify the genes, to locate the genes, and also to know the gene function. Those the distinctive types or the maps, they are called as, actually are two types. They are called as the genetic maps as well as the physical maps. Now, when you talk about the genetic maps, now genetic maps are nothing but the distances which are based on the genetic linkage phenomena or the information, and it is measured in centimorgans, that is cm. So it depends on actually the relative position of the loci based on the degree of recombination. Now this approach actually studies the inheritance or the assortment of the traits by genetic analysis, whereas the physical maps on the another side, they are actual physical distances between the loci, usually measured in the number of nucleotides or in the base pairs. So the physical map actually is more accurate, they say, when compared to that of the genetic maps or the genome maps. So what is genetic map or the genome map? Genetic map or the genome mapping is nothing but it is actually a method which is employed to find out the distance between the genes, the distance not only between the genes but also the distance which is present on the same chromosome but also on the different chromosome, and also to locate the position of the chromosome, as also to know the function of the chromosome or the function of the gene. So there are two types of these maps which is used. One is called as a genetic map and the other one is called as a physical map. Genetic map actually is based on the genetic linkage information, which is always measured in centimorgans, and it actually depends on the position of the loci based on the degree of recombination. Whereas physical map actually use the physical distances that are present between the loci. Usually it is measured in the number of nucleotides or the base pairs. So they say that actually physical map is more accurate representation, whereas the genetic map is not that much that accurate. Okay. Now coming to the relationship between the genetic map as well as the physical map. So we say that actually just now I've told that the genetic map uses what, a centimorgan. So 1 centimorgan actually on a chromosome is equal to 1 megabase. That is 1 megabase is equal to 10 to the power of 6 base pairs of DNA. But actually they say although the genetic maps of human females average 90% larger than the same maps, what do? Is their chromosomes contain the same number of the base pairs. So their physical maps actually are the identical. So that is what they infer. Now, so here, so genetic maps are the maps which are based or measured in centimorgans. Remember that. And physical maps are nothing but the physical distances which are measured in terms of nucleotides or the base pairs. Now, this is the difference between the physical and the genetic map they are given. And the physical map and the genetic map are not the same. They are totally different. Physical map will give you only the distribution of the genes based on the sequence data. That is based on the nucleotide base pairs. Whereas genetic map actually gives you based on the recombination frequencies. So that is why here, and these recombination frequencies are measured in terms of centimorgans. So the more closer together actually means the more, what do you say, the recombination frequency between them will be very low. And if they are very far off, then there is a high recombination frequency between the genes. This is the difference between the physical map as well as the genetic map. Now, basically, genetics use three different of maps to describe the location of particular gene. So no doubt, actually, we are talking about the physical map as well as the genetic map. Now, apart from this, in those days, genetics used to actually use three maps to describe the location of a particular gene on the chromosome. One type of map actually uses the cytogenetic location to describe a gene's position, where actually the gene is located in the what do you say, the location. And this cytogenetic location actually is based on the presence of the bands, distinctive bands, actually that the gene creates when the chromosomes are stained with certain chemicals. Another type of map which we call as the linkage map or the genetic map, just now I have mentioned, no? So that linkage map or genetic map is based on recombination frequencies. And here, the unit of distance in the linkage map is actually called as is equal to one map unit or one centimorgan. So one centimorgan or one map unit or linkage map is equal to nothing but 1% recombination. And the third type actually is called as the physical mapping. It is it actually tells you the position of a gene on the chromosome. So these are the three different types. Three different types. Actually, nowadays only they are using the physical mapping and the genetic mapping, but cytogenetic mapping they are not using, because here you need to actually use the certain dyes to stain the chromosome. So based on the banding pattern that these chromosomes give, then they will try to find out the cytogenetic location. Okay. So basically speaking, some of the scientists actually they say there are three types of maps. Some of them they say there are two types of maps. But if we as a thumb rule, if we see actually basically if we talk about, we can say that actually there are three maps to describe the location of a particular gene. One is the cytogenetic map or cytogenetic location. Second one is the linkage map or the genetic map, and the third one is the physical map. So remember, cytogenetic location or the cytogenetic map describes the position of the gene on a chromosome. And this cytogenetic location actually, how do we get this cytogenetic location? By trying to stain the chromosomes with certain dyes, and based on the banding pattern that they give, then the cytogenetic location or the site of that particular chromosome or the gene can be identified. Another type is the linkage or genetic mapping. So it is based on recombination frequency. So by recombination frequency. So here the unit of distance in the linkage map actually is called as the one map unit. So one map unit is equal to 1 centimorgan. Actually, why this is called as 1 centimorgan? Because this is given after the scientist name, that is called as Morgan. So one map unit is equal to how much? 1% recombination. And the third type actually uses the molecular location, that is physical mapping, where it will try to give you based on the molecular base phenomena. It will try to locate the position of a chromosome. So the molecular location actually is based on the sequence of DNA building blocks, that is the base pairs that make up the chromosomes. Okay, now we'll see one by one how actually these mapping techniques are being used, and how actually the scientists use these different techniques to locate the position of a gene, function of a gene, as well as the different aspects. Now coming to the cytogenetic mapping, that is called as cytogenetic location. Now this is actually, they say it is a standardized way to describe a gene cytogenetic location, because here the location here is based on the distinctive banding pattern of the stained chromosome. So here what we need to take, what the scientists actually has done is, they have taken the chromosomes and they have used certain dye, that is a chemical, to stain it. So when once these chromosomes have taken up the stain, they will show certain distinct bands. So based on this distinctive banding pattern, they could locate or they could identify this, the chromosomes or the location of this chromosome. So the combination of numbers and the letters, they provide a gene's address on a chromosome. So how they identified? So when once they could stain it, and when once they got the distinctive bands, they used several the mechanisms or the parts to describe actually where the gene is located. Say for example, suppose the chromosome on which the gene can be found from chromosomes 1 to 22, they are designated by their chromosome number. So the set chromosomes are designated by X or Y. So you need to follow certain rules. What is that rule is? You all know actually we have got 22 chromosomes, that are those are the autosomes, and one sex chromosome, that is the allosome. So now first you have to find know the chromosome 1 to 22. So they are designated by the chromosome number. So actually we number the chromosomes from 1 to 22. So the sex chromosomes are designated as X and Y. So this is one thing that we should know. Second thing is the arm of the chromosome. You all know each chromosome is divided into two arms or two sections based on the location of the centromere. Centromere, you all know, that is also called as kinetochore, which holds the two arms of the chromosome. So first we should name, or what do you say, give the number to the chromosome, that is ranging from 1 to 20. That is, those are all somatic chromosomes or autosomes, and the sex chromosomes, that is X chromosome as well as Y chromosome. Then the second thing is you should also know the arm of the chromosome. So there are two arms. One arm is a short arm and the other arm is a long arm. So based on that, that also we should find out. So here what we need to do is, the shorter arm actually you should give the name as small p, and the longer arm actually should be given as small q, or you can identify, you can actually give the name shorter arm as p and the longer arm as q. So the chromosome arm in the second part of the gene's address, this one. So for example, suppose, so if we actually, suppose if you want to identify one chromosome, you have taken that chromosome and you try to actually stain it. So you got certain distinctive bands. Now based on the address, you have to find out which particular chromosome it is and where it is actually located. Now say for example, they have given 5q. Now what does it indicate 5q? 5 actually stands for the chromosome number and q stands for the chromosome arm. So q is the long arm or short arm? Q is the long arm, whereas P is the short arm. So 5Q actually represents what? That it is the long arm of chromosome 5. Similarly, XP, capital X and small p. X is what? X is actually nothing but the X chromosome. And P is what? Nothing but the small shorter arm of the chromosome. So XP indicates the short arm of X chromosome. So like this, you have to identify. So this is one, what do you say, thing you need to follow. Then second thing is the position of the gene on the Q or Q arm. So where actually the gene is located on the P or on the Q arm. So this you have to find out. So the position of a gene is based on a distinctive pattern of light and dark bands that appear when the chromosome is stained in a certain way. So here actually the position is usually designated by two digits, which are sometimes followed by a decimal point or one or more additional digits. Say for example, now you have 14q21. Okay, and you want to find out, you want to know which chromosome it is and where actually it is located. In the initial stage, what we have seen the what is the chromosome number and whether it is a short arm or the q arm. Based on that, we have identified that this is the short arm of so and so chromosome or long arm of so and so chromosome. The second aspect what we need to identify here is the position of the gene. Okay, where actually the gene is located, whether it is located on the P or whether it is located on the Q, and also we have to designate it by two digits. Okay, like say for example 14q21 is there. Now what does it represent? It represents the position 21 of the gene on the long arm of chromosome 14. So 14q21, these two digits, that is the last two digits that you have given, that is 21, will represent the position 21 of the gene. It represents the position 21 of the gene on the long. Q stands for long arm. So position 21 of the gene on the long arm of the 14th chromosome. 14 is what? Chromosome number. So like this. The second aspect, if you see, it is 17q, 12q, 21. So it represents what? A range of bands. It represents a range of bands, okay, between 12 to 21. Okay, between 12 to 21, if less is known about the exact location of the gene on the long arm. So Q stands for what? Long arm. So it is a range of bands, okay, between 12 to 21, okay, on the long arm of the chromosome 17. So that is how actually you have to identify. So the number here indicates the gene position. It indicates the gene position, and this gene position will go on increasing with the distance from the centromere. So that is how you have to identify. This is with reference to the cytogenetic mapping. Okay. Now the fourth aspect is what? Now the abbreviation. They use the abbreviation either CEN or TER or TEL. Now they are actually used to describe a gene's cytogenetic location. They are used to describe a cytogenetic location. Say for example, CEN indicates what? That the gene is very close to centromere. Gene is CEN, centromere. Centromere. So it is a short, we can say it is a short form of centromere. Second one is what? TER. TER what? It stands for terminus. Now which indicates that the gene is very close to the end of the, very close to the end or very close to the terminus. Terminus. Then of the Q arm or the Q arm. Then TEL, what? TEL stands for the telomeres. Now, which are at the ends of the each chromosome. So that is how you have to identify. So, for example, if you have 16p7, okay, 16 is what? It is a chromosome number. That means chromosome 16. P is what? P is the represent the short arm, and sen is what? Nothing but centromere. So we can say that it refers to the short arm of chromosome 16 near the centromere. Near the centromere. So these are the different abbreviations that we use. One is sen or ter or tel. Sen is near the centromere, ter is near the terminus, and tel stands for the telomere. So like that you have to identify and you have to say it. So this is with reference to cytogenetic mapping. Now what does cytogenetic mapping say? Cytogenetic mapping is one type of gene mapping where by using this particular mapping, you can locate the gene which is present on the chromosome, cytogenetic mapping. So here what is you have to use, follow certain rules. You have to number the chromosome from 1 to 22, sex chromosomes as X and Y. Then after that, you have to also designate the names like Q as well as P. Now you all know actually you have got the, each chromosome has got a centromere in between, and this centromere holds the two chromatids. So one is a short arm and the other one is a long arm. So P actually we use it for short arm and Q is a long arm. And another is we have to use an abbreviation like CEN, TEL or TOR. So based on that, we can know where the chromosome is actually located, whether it is near the centromere or whether it is near the telomere or whether it is near the terminus, means close to the terminus or close to the centromere like that. So this is how you have to find out or locate the gene on the chromosome. So this type of mapping or gene mapping, mapping is called as nothing but the cytogenetic mapping. Okay. So this is how, see here we have given. See, this is banding pattern. That is how you do. So this you will get it only in case of cytogenetic mapping, but not in the physical mapping, but not in the linkage mapping or the gene mapping. Because here in the cytogenetic mapping, what do? Is they stain the chromosomes. Then once they stain the chromosomes with a certain dye, they do like G banding, Q banding, R banding, like different types of stains you have. So when once you stain it, the chromosomes actually will take up the what do you say, banding pattern. They'll show the banding pattern. Okay, dark and light bands, and characteristic banding will be seen. So like that, based on this, you can actually give the number. Okay, you can indicate the short arm and you can indicate the long arm. You can indicate the numbers of the chromosomes as 1 to 20, 22 as well as X and Y chromosome. And also you can use the abbreviation for centromere, for terminus as well as for the telomere. So based on that, you can actually find out the location of the chromosome. So this is how you do it for cytogenetic mapping. Okay. Now this is the chromosomal location of a gene. See here, they have seen. Example they have given. This is the long arm, this is the short arm, you all know actually. Now this is the centromere, which is located in the center. Okay, this is the region of 3. Okay, yeah, banding pattern is there. See, this is the band 1, this is the sub band 2. So like this, and this is the chromosome 7. So based on that, you can actually locate the chromosomal location of gene, where actually the gene is located like this. Okay. The next is what? Genetic mapping. Now, genetic mapping is nothing but a gene mapping. Now, the gene mapping actually is based on the recombination phenomena. Actually, it is given by Morgan, I told you, and it is measured in units. One map unit, or you can say one map unit is equal to 1 cm. That is centimorgan. So that centimorgan, because it is based on the recombination frequency, 1 centimorgan is equal to 1% recombination frequency. So that is how actually you go about with the gene mapping or the genetic mapping. And the third one actually is the physical mapping. Now, the physical mapping actually involves the breaking the chromosomes into several fragments, DNA fragments. So what do? Is in physical mapping, they try to break the chromosomes into smaller DNA fragments using different types of techniques so that they can be handled individually and you can order them, okay, into fragments to their respective locations on the chromosomes. Since you don't, actual base pairs are generally very hard or impossible to measure, the physical maps actually are constructed by first shattering the genome into small pieces, then by characterizing each single piece and then assembling. Again, you have to actually break them and again assemble them back, okay, into an order. Then you have to overlap path or tilting path of these small fragments would actually allow the researchers to infer physical distances between genomic features. So that is how actually you try to know the physical map. Now, these are the three different types of the, you can say, the gene mapping or the genomic mapping methods. Yes, students, are you all listening? Hello? Hello? Hello?
Yes, ma'am.
Uh, Okay. So this is how actually the gene mapping techniques actually will work. So how many gene mapping methods are there? Three mapping, basically three mapping. But now the scientists actually are not going for the cytogenetic type of mapping, because there you have to actually stain them and then you have to identify. But basically speaking, as a rule, if we see, there are three different types: cytogenetic mapping, then you have got the genetic mapping and the physical mapping. Cytogenetic mapping involves what? It involves the what, staining of the chromosome with a dye, and then when once the distinctive bands come, then what you have to do? You have to assign the numbers to the chromosomes, autosomes as well as X chromosomes, and also you have to name the short arm as well as the Q arm. That is Q and P. You all know one is the long arm and the other one is short arm. And also you have to, if you want to exact find out the location where actually it is being present, then you have to give abbreviation like in the form of CEN, TEL, or TER. CEN stands for centromere, TEL stands for telomere, and TER stands for the terminus. So that way you can identify. Next is what? Next year genome mapping, that is the genetic mapping. So it requires actually a number of base pairs. So based on the base pairs, you have to find out. That is, recombination. So map unit, each single map unit, and it is one map unit is equal to 1 centimorgan. And I told you the Morgan, why it is called as centimorgan, the unit name is, it has been given after the scientist called as Morgan, and 1 centimorgan actually is equal to 1% recombination frequency. So it is actually based on the recombination phenomena. And physical mapping, you all know what we need to do is, here in the case of physical mapping, they break the chromosomes into small fragments, because you cannot handle the chromosome. I mean, because chromosome structure is very, very big, the size is very, very big. So since you cannot handle them, so what they do is they try to break them, okay, by means of different methods like fragmentation and all those things. They apply different techniques, and then they try to break that chromosome, okay, into small fragments. And after breaking them, you have what do they do is they actually arrange them again into pieces. Means they'll assemble back. First they break them and then they assemble back together, and then they will try to find out or know the physical distance between the chromosomes. That is what is called as the physical mapping. Okay, see here. Now, just now I've told you, know, the physical mapping, one of the method that they use is actually called as fragmentation. So this fragmentation, how do they do? You all know. You can break the chromosome, means you can dissolve the chromosome by the help of restriction enzymes. You all know restriction enzyme actually is an important enzyme which will cleave or which will cut the DNA into small pieces. Okay, so that is how what they do is they take the restriction enzyme and then they try to cut this DNA, okay, or through shattering it by sonication, or either they give a shear, what do you say, stretch by sonication phenomena, and then they try to break the DNA. So when once the DNA is being cut or when once the DNA has been shattered, then the DNA fragments actually forming the nucleus, they are assembled, okay, by either automation, that is by machine, or by manual means into overlapping DNA stretches. Okay, so then they will have an observation. So now a good choice of the closes can be made to efficiently sequence the close to determine the DNA sequence. So that is how actually you can do it. Now, but actually, you know, the genome sequencing has one major limitation. Now, even with the most sophisticated technology, why? Because it is rarely possible to obtain a sequence of more than about 750 base pairs by a single experiment. So you cannot get more than, and you all know the length of the DNA is very, 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 very big. So you cannot actually do it at a time. So what they say is it is highly impossible to obtain a sequence. So you can get it only about 750 base pairs in a single experiment, but you cannot get more than that. So this means that the sequence of the long DNA molecule has to be constructed through a series of short sequences. So you have to go on actually cut it and then go on doing the experiment. So then the next what they have done is they have shotgun sequencing method actually is the standard approach for sequencing, they say, for small prokaryotic genomes, but it is much difficult with the larger genomes because the required data analysis becomes more complex as the number of fragments go on increasing. Then the second problem with this method is what? It can lead to more errors because the repetitive regions of the DNA of a genome will be analyzed. So that is the reason why actually these are some of the drawbacks. So whenever you are doing it, you have to be very, very careful because the DNA length is very, very much. So it is very difficult to get the whole DNA sequencing in a stretch. So that is the reason why they go for the sequencing by following the different methods. So in small pieces, in small fragments, if you do it, then it will be more accurate and it will give you the correct result. So that is what these scientists actually have suggested. Okay. The next this is now benefits of physical mapping. So, based on all these things, since they have said that there are three methods of genetic mapping or the genome mapping, one is cytogenetic mapping, gene mapping, as well as the physical mapping. Among these three, they say that physical mapping actually is more beneficial. Physical mapping is more beneficial when compared to this. Now, we'll see why. Because a genetic map is used to locate and identify the gene or a group of genes that determines a particular trait. So here the techniques that are developed for genetic mapping actually have a great impact on life sciences as well as medicine. So, but genetic mapping technologies also have applications in other fields also. So a roundup of genetic mapping applications include what they have given here. One is called as medicine, agricultural applications, energy and the environment. So actually these mapping methods or the mapping technologies actually have been used for the genome sequencing or the genetics. That is the actually purpose of this gene mapping. So why actually they require a gene mapping? Nothing but to sequence the genes or to when once you sequence the genes, you can know the structure of a gene in to a full stretch. You can know the function of each gene and where actually the location of each gene, where actually a gene is located. You can identify how many genes are there, what are these genes, where are they located, what is their function, what is their role, and which gene is responsible for which type of character, and which gene is actually will cause which type of disease. So that is why sequencing for the purpose of sequencing, this genome mapping or the genetic mapping actually have been identified or actually have been given by the scientists. So now this genome mapping or the genetic mapping actually is more beneficial in genetic sequencing. So it is used in medicine, it is used in agricultural applications, it is used in the energy as well as environment. So you can know actually you can sequence the gene. So that is the main purpose of this what do you say, genome mapping or the gene mapping. The next you can also use in forensics. Now, for more detectable applications, gene mapping can also authenticate the what origins of the consumer goods, the like fruits, wine, or the other thing. Okay, so for this, actually they use the genome mapping or the genetic mapping. Is it okay, students? Hello, hello.
Yes, ma'am. Yes, ma'am.
So gene mapping or genome mapping is one and the same. So what we can do is why we have to go for mapping the gene. What do you mean by mapping the gene? Nothing but finding out the position of a gene on a chromosome. So when once you find out the position of a gene on a chromosome, you can know how many genes are present on a chromosome, what are they, which gene is responsible for which character, which gene actually causes what type of disease. So you can sequence the gene, you can know. Okay, and also you can have it has got wide applications. You can treat the diseases. So in medicine, in forensic science, you can know. So that is the reason why this gene mapping is very, very important. So these scientists actually, when they try to go for DNA sequencing or the genome sequencing, they could identify three types of gene mapping methods. One is called cytogenetic mapping, second is the genetic mapping, and the third one is the physical mapping. Cytogenetic mapping is based on what I told you. It is based on nothing but the staining. So the chromosomes actually need to be stained by using a certain dye. So when once you stain it, it shows distinctive bands. Now to identify which gene it is, you have to be very careful. So you know, for example, human chromosomes are 23 pairs. Among them, 22 are autosomes and one is a sex chromosome. So you have to number the chromosome from 1 to 22 for somatic and X and Y for that. Okay, after that, what I told you, you have to actually each chromosome, since it consists of two chromatids, that is arms, one is a short arm and the other one is a long arm, you have to designate the symbol as small p and small q. Then where these chromosomes are located. Suppose you have identified that gene, but that gene is located where? Whether it is near the centromere or whether it is near the terminus or whether it is a telomere. For that, you have to use an abbreviation. So like that, you can design or use these abbreviations and you can identify or locate the gene. That is what is called cytogenetic mapping. Second one we have seen, it is nothing but the gene mapping. Now it is based on the linkage or the recombination phenomena. So here, the more close they are means recombination will be very less. Okay, if they are very far apart, then the recombination will be more. So actually, it is based on the map unit. So one map unit actually is called as 1 centimorgan, and centimorgan is equal to nothing but 1% recombination. So that is how. So here you have to actually use the recombination frequency for the genetic mapping. And the last one is the physical map. Now, in case of physical map, what you need to do is you have to actually use different methodologies to actually cut the DNA molecule into small fragments and then assemble it back and then try to identify. That is by fragmentation phenomena. Because since DNA, what do you say, the size is very, very large, it is very difficult actually to sequence a DNA, whole DNA at a time. So in this method, what they try to do is they try to actually cut the DNA sequences with the help of restriction enzyme. Then once they got these fragments, all these fragments, they try to assemble it based on their base pairs. And then when once they assembled it, and they could identify the what do you say, the location of the chromosome. That is called. So among these three, they say that physical mapping is very, beneficial when compared to the two different types of the genome mapping or the gene mapping. Is it okay? So now the genome mapping or gene mapping has got wide applications. It is used in agriculture, it is used in forensic science, it is used in medicine, it is used in energy as well as in the environment. Okay? Is it okay? Yes, students?
Yes, ma'am. Yes, ma'am.
Okay. Now we'll go to some other topic. Okay? Just wait a minute. Okay. Now the next chapter that we will see today now is mutations. Yes, anyone is having any doubt regarding the gene mapping? Yes? What students? Hello? Anybody is having doubts? Hello? Okay, now coming to the next topic, that is mutations. Now we will see now what are mutations. Now mutations are nothing but the sudden changes in the genome sequence or the sudden changes in the gene. Now when we talk about the mutations, now mutation actually is nothing but a heritable change, you can say, or it is a process that produces a gene or the chromosome. Okay, that actually helps in the production of a wild type of chromosome. So here the mutation may result due to many changes. It may be either on the gene or either on the chromosome itself. So basically speaking, we talk about two different types of mutations. One is called as the gene mutation and the other one is called as the chromosome mutation. Now the gene mutation actually involves the changes in the gene, whether it is the allele of a gene or the whole gene as such. Whereas the chromosomal mutation actually involves the mutation or the changes in the chromosomes or in the segments of the chromosome, whole chromosome, or the entire set of the chromosomes. So that is how we designate two different types of mutation. So what is mutation? Mutation is nothing but a process. Actually, it is a sudden heritable change that produces a gene or a chromosome of a different type totally. And it occurs due to changes either on the chromosome or either on the gene or either on the chromosome. So broadly speaking, the mutations actually have been classified into two different types. One is called as the gene mutation and the other one is called as the chromosome mutation. Now, gene mutation, actually, in case of gene mutation, there will be change in either in the gene or either on the allele. Whereas in case of chromosomes, here it may involve the whole chromosome or the entire set of the chromosome or the only a segment of the chromosome. See, now coming to the types of mutations. So there are number of various applications you can see, or the various schemes for classification of these mutations. So based on different aspects, these mutations actually have been classified. One is the type of cell involved, whether the mutation you see it in the somatic cell or whether you see it in the sex cell. So based on that, first category is based on the type of cell involved. The mutations actually have been classified into different types. One is called as somatic mutation. Now, somatic mutation, actually, somatic soma means it refers to the body tissues. So mutations actually that are seen in the somatic tissues of the body, you call it as somatic mutations. And these mutations actually are not transmitted to the progeny. Why? Because it is not related with sex. So it is related only in your body tissues. So that is why they are not transmitted. And the extent of the phenotypic effect depends on whether the mutation is recessive or whether it is dominant. So it is generally have a very less effect. And the extent of the phenotypic effect also depends upon whether it occurs early or during late development. So that is what is called somatic mutation. Now, somatic mutation is what? It is nothing but the mutation, okay, which is observed in the somatic tissues of the body. They are not transmitted to the progeny. And here the extent of the phenotypic effect because of this mutation depends on whether the mutation is recessive. And also, it depends whether it occurs early or whether it occurs during the late development. So that is what is the somatic mutation. Okay, the next is what you have germinal mutation, you can say, or germ mutation. Now this is, okay, this is with reference to germ mutations or the germinal mutations. Here you require germ cells. Germ cells are nothing but the sex cells, right? So here mutations actually that are seen in the germ tissues of the body. So this may be transmitted to the progeny. You all know actually whatever the characters are there, they are being transmitted from the parent to the offspring. So here the mutations, suppose if it occurs in a parent, that will be transmitted to the offspring or to the progeny. So these type of mutations actually may be transmitted to the progeny. Now here the dominant mutations actually are seen in the first generation after the mutation occurs. Now if a female gamete contains an X-linked mutation, then what happens? X-linked mutation is fertilized, then the males will have the mutant phenotype. And recessive mutations will only be seen upon the chance mating. Okay, so this is what is about the germinal mutation, you can say, or the germ mutation. So the germ mutations or the germinal mutations are seen in the germ tissues of the body. They are observed in the germ tissues of the body and they are transmitted to the progeny, that is from parent to the offspring. And the dominant mutations actually are seen only in the first generation after the mutation occurs. Then this is the first category. Because of the based on the type of cell involved, you have two different types of mutation. One is somatic and the other one is germinal. Now, based on the mode of origin, you have two different types of mutation. One is called spontaneous mutation, and then you have got induced mutation. Now, spontaneous mutations occur suddenly in nature. They occur suddenly in nature and their origin is unknown. From where actually you got the mutations, from where actually it occurred, you don't know. So it occurs suddenly in the nature, and they are also called as background mutations. They are also called as background mutations, and they have been reported in many organisms like maize, Enothera, bread mold, and microorganisms like bacteria as well as viruses, even Drosophila, mice, even man. So such mutations you call spontaneous, means which occur very suddenly. You don't know actually from where it has occurred, what is the background and all those things. So that is why it is called as a spontaneous mutation. Means it occurs naturally. That is what we say. Then next is what? Induced mutation. Now, besides naturally occurring spontaneous mutations, the mutations can also be induced. Okay, so in one way we can call this as artificial mutations. Whereas because we are trying to induce, we are trying to actually provoke to have the mutations. But here in case of spontaneous, we are not doing. Whatever the mutation that occur, it occurs naturally. Okay, without any induction. But in case of induced mutations, what we are doing? We are trying to actually induce the mutations. okay, artificially by exposing the chromosomes to different abnormal conditions, abnormal environment, such as you may expose it to radiation, you may expose it to temperature, chemicals like X-rays, gamma rays, and all those things. So here you are trying to induce the mutations, you are trying to create the mutations by with the help of certain environmental factors. Okay, by exposing to radiation, UV rays, say gamma rays, or chemicals, or temperature, or whatever may be the reason. So in one we can say spontaneous mutations, you can call it as natural, whereas induced mutations, you can call it as artificial. Then the third category is based on the direction of mutation. The mutations can be classified into two types. One is called as the forward mutation, and the other one is called as the reverse or back mutation. Now, forward mutation is when the mutations actually create a change from the wild type to the abnormal phenotype. When the mutations create a change from a wild type to an abnormal type, then these mutations you call it as forward mutations. So most of the mutations actually are forward type. They do not come back. So you're going forward. Now reverse or back mutations is here the forward mutations are often corrected by error correcting mechanism so that an abnormal phenotype changes into a wild type phenotype. So it is a reverse. In forward, you always create a change from wild type to the abnormal type. But here, what is from abnormal type to the wild type you are creating. So this is the reverse or the back mutation. The next category is what? Based on the size and the quality. Okay, you have two types of mutations. Based on the size, okay, two types of mutations actually have there. One is called as the point mutation, and the other one you call it as the multiple mutations or the gross mutations. Now coming to the point mutations. Now if you see the point mutations, the point mutation is when heritable changes occur in a very small segment of the DNA, very small segment. It involves a very small DNA segment of the DNA molecule, or either maybe a single nucleotide or a nucleotide pair. Then this type of mutation you call it as the point mutation. Now the point mutations occur due to following types, okay, of subnucleated change in the DNA or in the RNA. So they may be due to deletion, insertion, or substitution. So point mutations actually occur in a very small fragment of the DNA, a small segment of the DNA. It does not involve the whole nucleotide. Only a small molecule or small segment of the DNA molecule or a nucleotide or nucleotide pair. Okay, and this occurs due to the changes either in the DNA nucleotide, means subnucleotide change either in the DNA or the RNA, and it is of different types like deletion, insertion, or substitution. Now when you talk about deletion, the point mutation which is caused due to loss, deletion means loss, so which is caused due to loss or deletion of some portion, okay, in a triplet codon of a cystone or a gene, it is called as deletion. The next is what? Insertion mutation, or insertion means you are adding something. So here insertion or addition mutation. So the point mutations which occur due to addition or more extra nucleotides. So when more extra nucleotides are being added, okay, then to a gene, then it is called as insertion mutation. And these mutations arise from the insertion or deletion of individual nucleotides, and then they cause the rest of the message downstream of the mutation to be read out. Then you have substitution mutation. Now, any point, a point mutation actually in which a nucleotide of a triplet is being replaced by another nucleotide, then such a mutation you call it as a substitution mutation. So we can say based on the based on the size, there are two different types of mutation. One is called point mutation, and the other one is called as the gross mutation. Now, the point mutation actually involves a very small fragment of the DNA or the RNA, okay, and it occurs either due to deletion, insertion, or substitution. Deletion is loss, insertion is when we are incorporating, when you are adding, and substitution is when actually a point mutation, when in which a nucleotide of a triplet codon is being replaced by another nucleotide, then you say it is substitution. Then second one is multiple mutations or gross mutations. So when the changes occur in more than one nucleotide pair, or say for example an entire gene, then such mutations you call it as gross mutations. So there in point mutations it was involving only a small segment. Okay, but here it involves a whole one nucleotide pair or the entire gene. Then you call it as a multiple mutations or the gross mutations. And these gross mutations you all know actually it occurs due to arrangement of genes within the genome. So these the genes will take that what do you say? They'll try to shuffle, they'll try to shift their places and all those things. So due to the rearrangement of the genes within the chromosome, the gross mutations actually occur. So the rearrangement of genes may occur within a gene. So it may occur as a gene. So two mutations within the same functional gene can produce different effects depending on the gene, whether they occur on the cis or transposition. Then the arrangement of gene may occur in a number of genes. First is within a gene, it may occur, or the rearrangement of genes may occur in a number of genes per chromosome, and due to the movement of a gene locus also, new type of phenotype also may be created. That in that way, actually these multiple mutations or the gross mutations are created. So it is because of, since it is because of the rearrangement of gene which takes place either within a gene, means the rearrangement of gene may take place or may occur either within a gene or in the number of genes per chromosome, or it may be due to the movement of a gene locus occurs, then the multiple mutations or gross mutations are created. Okay. Now again, these are different types. Like, okay, one is called translocation. Now translocation is what? Nothing but movement of a gene may take place to a non-homologous chromosome. This is called as translocation. They are of different types. One is translocation and the other one is inversion. Now translocation is movement of a gene to a non-homologous chromosome is called translocation. Inversion is the movement of a gene within the same chromosome. You call it as inversion. Okay, so this is about the multiple mutations. Okay, now next, based on the phenotypic effects, based on the, you know what is phenotype? Phenotype is nothing but the external appearance. You call it as a, or external expression of a gene, you call it as a phenotype. So here, phenotypic effects. Based on the phenotypic effects, now you have different types of mutations. One is called as the morphological mutations, and the other one is lethal mutations. Then you have got the control mutations and biochemical mutations. So these are the four different types of mutations based on the phenotypic effects. So what are those? One is the morphological mutation. Now morphological mutations actually are the mutations that affect the visible properties, whatever the properties that we can visualize that affect the visible properties of an organism externally or morphologically, then we say it is morphological mutation. For example, curly ears in cats. So like that. So morphological, whatever the part of the body that is visible outside, then it is called as morphological mutation. Then you have lethal mutations. Now, lethal mutations actually are the mutations that affect the viability of the organism. Lethal mutations are what? They are nothing but the mutations that affect the viability of the organism. Then conditional mutations are the mutations in which the mutant allele will cause the mutant phenotype only in certain environments. That is what is called condition. Means only under certain conditions, only under restricted conditions. Okay, they are being formed. So, or they occur. So, conditional mutations are the mutations in which the mutant allele causes the phenotype only under certain environment or only under certain specific conditions. So, this is called as the conditional mutation. So, then next you have biochemical mutations. Now, biochemical mutations actually are the mutations that may not be visible or affect a specific morphological characteristic. Morphology means externally it is visible, but biochemically it is not visible externally, but they have a general effect on the ability to grow or proliferate. So it actually causes changes or the alterations in the biochemical parameters. So as a result, what happens? They'll have an impact or the effect on the ability to grow as well as to proliferate. So this is called as the biological mutation. Now the next category is what? Based on the magnitude of phenotypic effect. Based on the magnitude of phenotypic effect, now you have different types of mutations, that is dominant mutation, recessive mutation, and isoallele. Now coming to dominant mutation, you all know dominant. What is a dominant? It is nothing but which is actually expressed always. So the mutations which have dominant phenotypic expression, the mutations which have a dominant phenotypic expression, they are called as dominant mutations. For example, in man, the mutation disease, anidia, that is absence of iris of eyes, occurs due to a dominant mutant gene. Then recessive mutations. Now, most of the mutations are recessive in nature, so they are not expressed immediately. You all know dominant will be expressed, okay, but recessive will not be expressed always, and they will not be expressed immediately. So the phenotypic effects of mutations of a recessive gene is seen only after 1 or 2 generations, but not in every generation. So such mutations, you call it as the recessive mutation. And the third one is isoalleles. So, some mutations actually alter the phenotype of an organism so slightly that they can be detected only by special techniques. So, the mutant genes that can be slightly modified phenotypes, they are called as nothing but the isoalleles. So, these isoalleles, what do? They produce identical phenotypes in homozygous or heterozygous condition. So, this is with reference to the magnitude of the phenotypic effect. You have three types of mutations. Now, next category is whether based on the laws of function or the gain of function, how the mutations actually are being classified, we will see. Okay. Now, first is based on the laws of function. So, laws of function mutation. So, based on the laws of function and gain of function mutation. So, laws of function mutation is also called inactivating mutation. Inactivating mutation, which results in the gene product having less or no function at all. So here the word itself indicates that there is a loss of function. So this type of mutation actually is an inactivating mutation. So it will result in the gene product having very less function or having no function at all. Okay, it may be either partial or either it may be totally inactivated. So such a type you call it as the loss of function mutation. Now gain of function mutation actually are called activating mutation or they are active mutation. So what do? They change the gene product such that its effect will get stronger or even is suspended by a different and abnormal function. So that is what is called as the gain of function mutations. So loss of function mutation and uh, 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 loss of function mutation and gain of function mutation. In case of loss, what happens? They are inactivating, whereas in case of gain, they are activating mutations or the active mutation. Now, another category is what the type of chromosome involved. So based on the type of chromosome involved, the mutations actually are classified into different types. Now, what are those we will see. Now, here the chromosome autosomal mutation and sex chromosomal mutation. So if the mutation is seen or occurs in the autosomal chromosomes or the autosomes, such mutations you call it as the autosomal mutation. And if the mutations actually are seen in sex chromosomes, then you say it is called as a sex chromosomal mutation. Now, autosomal mutation, they are autosomes from 1 to 22, whereas sex chromosomal mutations actually are X and Y chromosomes. So, this type of mutation, it occurs in the sex chromosomes. Now, we will see one by one what are the different types. Now, first is chromosomal mutation and the types. Now, the changes in the genome involving chromosome pairs, okay, they are called as a chromosome mutation. Now, this involves a number of things. It may be due to changes in its structure, or it may be not only in the structure but also in the size of the chromosome or shape of the chromosome. So, based on the changes in the number of genes, or changes in the arrangement of the genes, or changes in the number of the chromosomes, actually, you have different types of chromosomal mutation. So, they have been divided into different categories of these chromosomal mutation. Now, one is structural changes in the chromosome. So there may be, see, whenever a mutation occurs means there is a sudden change in the chromosome, whether it may be with reference to number or whether it may be with reference to in the arrangement of genes or whether in the number of the chromosomes like that. Okay. So first is the structural changes in chromosome. Now, changes in the number of genes. Okay. It involves what? Deletion as well as duplication. Deletion or duplication. Now, changes in the number of genes. Deletion is what? Loss. So deletion involves what? Loss of a broken part of a chromosome. Either it may be a one gene or the whole gene. What? Addition is what? Addition is nothing but duplication, which involves addition of a part of a chromosome. It is also called duplication. So here, changes with reference to number of genes actually are of two types. One is called as a deletion and the other one is duplication. Now the second one is changes in the gene arrangement. Now changes in the gene arrangement is like, say for example, rotation. You have now, when the gene undergoes rotation, or if there is a change in its shape or in its structure, then rotation actually. So rotation is what? Rotation of a group of genes through 180 degree within one chromosome. Then you say it is a rotation. Then another mutation is inversion. Inversion in which the broken segment reattached to the original chromosomes in the reverse order. Then you say it is inversion. So like this, actually you get different types of what do you say, the changes in the gene arrangement. So changes in the gene arrangement are of two types. One is called rotation and the other one is called inversion. Rotation actually is nothing but it is a rotation of a group of genes, okay, around 180 degrees within one chromosome. And inversion is what? It is nothing but the arrangement of what do you say, broken segments, you can say, are again reattached to the original chromosome in a reverse manner. Then next is what? So this is one thing. So rotation or inversion means one and the same. The next is what? Exchange of parts between the chromosomes, okay, which is called as translocation. Exchange of parts between the chromosomes of different pairs. So translocation is what? Translocation is nothing but in which the broken segment will become attached to a non-homologous chromosome. So they try to exchange their parts by becoming attached to the non-homologous chromosomes, resulting in a new linkage relations or in new linkage groups. That is what is called translocation. So this is with reference to structural changes in a chromosome. Now coming to the mutations with reference to changes in the number of the chromosome. Okay, so suppose there is a change in the number of the chromosome. Instead of 23 pairs, that is 46, we may have 47, or we may have less, that is 45 or 42, like that. So that will result in the mutation. So changes in the number of the chromosomes. Now changes in the number of the chromosomes, okay, are of different types. First one is called as euploidy. Okay, there are two types. One is called euploidy and aneuploidy. Now, if you talk about euploidy, what? It involves either the loss or gain of the whole chromosome set. Okay, it involves either the loss or gain of the whole chromosome set. So the term euploidy, eu means actually true, ploid meaning unit. So it designates the genomes containing chromosomes that are multiple. So the euploids are those organisms that have a balanced set of chromosomes. So the number of chromosomes in a basic set, you call it as a monoploid number. And these monoploid whose number of sets is greater than 2, then you say it is polyploid. So if you have one set, you say monoploid. If you have 2, you say it is diploid. If you have 3, then you say it is triploid. If you have more than 3, then you say it is polyploid. Then if you have 6, then you say it is tetraploid. Fifth is pentaploid. Tetraploid is for fourth one. Then hexaploid for 6, and so on. So overall together, we can just call it as the polyploid. So the mutation due to euploidy refers to what? Nothing but the state of having a chromosome number that is an exact multiple of the basic chromosome. So here in euploidy, what is happening? It is the number of chromosome is increasing. That is what we call it as euploidy. So it involves either the loss or the gain of the whole chromosome set. So based on this, they are classified into two types. One is you have polyploidy. We'll see now. Polyploidy is addition of one or more sets of chromosomes. So this polyploidy is of two types: autopolyploidy and allopolyploidy. Autopolyploidy involves polyploidy in which the same basic set of chromosomes actually are being multiplied. Auto means same. Allopolyploidy actually results due to doubling of the chromosome number in the F1 hybrids, which are derived from two distinct species. So that is what is called as the alloploidy. So polyploidy, and so euploidy is what? Either the loss or the gain of the whole chromosome set, and it refers to the chromosomes that are multiples or basic. They are the organisms which are balanced set of chromosomes. The number of chromosomes actually in a basic set is called as monoploid. If you have 2, you say it is diploid, 3 triploid, then 4 tetraploid, 5 tetraploid, hexaploid, and so on. If you have more than that, then you say it is polyploid. This polyploid is of two types: autopolyploidy and allopolyploidy. Now coming to the next, that is aneuploidy. Now aneuploidy involves the loss or gain of a part of a chromosome set. Here, whole chromosome set it was in case of euploidy, but in aneuploidy, it involves the gain or loss of a part of the chromosome set. So it refers to a condition in which one or a few chromosomes are added or deleted from the normal chromosome number. So here, hence, the number of the chromosomes actually can be either greater or smaller than the chromosome. So various types of aneuploidy actually are there. That is nullisomy, monisomy, monosomy, trisomy, like that. Okay. So like this you have. So aneuploidy is what? Loss or gain of a part of the chromosome set. Now nullisomy refers to what? Loss of both the chromosomes of a homologous condition, that is 2 and -2. So it may be lethal in most of the organisms. Then monosomy is what? Loss of only one single chromosome. Then trisomy is what? It is a gain of an extra chromosome, that is 2n plus 1. It is 2n plus 1, where you have extra chromosome actually is being added. Instead of 46 chromosome, okay, you may have 47 chromosomes. So like this. So aneuploidy refers to the loss or gain of a part of the chromosome set. So various types of aneuploidy are available. One is allosomy, monosomy, as well as the trisomy. Okay. Then next is what you have different types, like you have missense mutation, nonsense mutation, and the frame shift mutation. Now, what do you mean by missense mutation? Now, missense mutation actually is nothing but when there is a change in the base pair, means a base change that converts the one codon into another, then you call it as nothing but missense mutation. Now, these missense mutations actually are silent because the encoded amino acid will remain the same, or the amino acid substitution is sufficiently settled or is not compromised activity. Okay, so that is what is called as a missense mutation. Missense means any base change that will convert one codon into another, and these many missense mutations actually are silent. Then next you have nonsense mutations. Now nonsense mutations actually a base change that converts a codon within the coding sequence. So what it will try to do, a base change that converts one codon into another, but here it converts a codon within a coding sequence into a stop codon. It will stop. So now there is only a limited set of sense codons will be there, so that can stop the codon. So that is why they are called as the nonsense codons or nonsense mutations. And the last one is what? Frame shift mutation. Now the addition or deletion of either a base or bases such that the coding sequence is shifted out of register or shifted out of place, then you say it is frame shift mutation. Totally it is shifted. So here, so either the addition or deletion of a base or bases such that the coding sequence is being totally shifted out of the sequence. That is what is called frame shift mutation. So these are the three different, what do you say, the apart from these are the three different types of mutation. So now we have seen what is mutation, what are the different types of mutations based on different criteria, and you all know mutation is what? It is a very, very important aspect. Mutation is nothing but a sudden change in the gene, either in the complete gene or either in a part of a chromosome or a segment of the chromosome or a part of DNA. Or so based on this, these mutations actually have been classified, and there are several criteria actually that I mean, based on that, these mutations actually have been classified. Okay. Hello. Is it okay, students? Hello.
Yes,
Yes.
ma'am. Yes. Yes, ma'am.
Okay. Now we will. So, any doubt is there in the mutations? Yes.
We will refer the book, ma'am, once and.
Okay. Now I'll talk a little bit about the chromosomal disorders. Is it okay? What are
Okay.
chromosomal disorders?
Okay, ma'am.
Just wait a minute. Okay. So I'll give you a very gist of the different types of chromosomal disorders actually that are often seen. So you all know actually we have seen now just now what are mutations, what are the different types of mutations, why mutations occur. Okay, there are various reasons or factors responsible for causing the mutations. So these mutations may be either natural or they may be either artificial or they may be either due to environmental factors. So many reasons actually are there for the cause of the mutations. Now, because of these mutations, actually, it will cause various number of disorders, or you can say various numbers of aberrations. Now, these chromosomal disorders, or you can say the chromosomal aberrations, are all mean one and the same. So, how does these chromosomal disorders actually will take place? It is nothing but because of the mutations. Because mutations lead to changes. Either it may be a change in the number of the genes, or due to the structure of a gene, or so many reasons just now we have seen. So this definitely will lead to what? Either different types of what do you say, aberrations, or different types of chromosomal disorders, or different types of manipulations. You can see they all mean one and the same. Either it is they call it as either the chromosomal disorders or genetic disorders, or you can call it as genetic manipulations or genetic aberrations or chromosomal aberrations. They all mean one and the same. Now, I'll give you a few examples, okay, of these genetic disorders that are often seen, genetic disorders or the chromosomal disorders that are often seen. Now, one is the Down syndrome. You might have heard about Down syndrome. Now, Down syndrome actually has called as a trisomy 21. Now, trisomy 21 means it has got three copies of chromosome 21. Means trisomy, three copies of the chromosome 21. Instead, actually, everyone will have only one. But in trisomy condition, you have three sets of this trisomy, three copies of the chromosome 21. And it can be either a male or a female. And they are physically and intellectually challenged. They are totally challenged. Means they are physically challenged, means they are handicapped. Like. And the incidence actually occurs. I mean, the incidence in about 8,000 to 800 to 1,000 births, only one will be affected by this. And the most common non-lethal trisomy it is. It is not lethal. It does not cause any harm, but it is a non-lethal trisomy. And 95% of this disease, or you can say the syndrome, is due to maternal non-disjunction. It comes from the maternal side, and the remaining 5% actually will come from the mosaicism or from the translocation. And in majority of the cases, the egg will pass on the extra chromosome, and there are a number of cases where the sperm will pass on the extra chromosome. So here, what are the salient features? You can say the characteristic features of this syndrome means the people who have this syndrome is the affected. Usually, mostly the children are affected by this, and they have marked hypotonia, tongue protrusion will be there, and they have got a very small head, flattened occiput is there, and flat nasal bridge, epicanthal folds will be there, and then with up slanting palpebral fissures, loose skin around the neck, and they'll be very short. Their fingers will be very stubby and single. See here, like this. This is an example with trisomy 21. See here, you see, they have given over here. This is the example, and that is a karyotype. Okay, so this is all about the trisomy 21. So trisomy 21 example also you have seen. So this is the example. I mean, it is a Down syndrome which comes under trisomy 21. Okay. Now, another features are they have got the narrow eye openings will be there, up slanting eyes will be there, arched eyebrows will be arched, and they have arched palate, flat nose bridge, bow-shaped mouth will be there, and they have got very small ears, short neck, and then their sloping shoulders will be there. Associated defects include apart from these external features, they also have got what is a endocardial cushion is there, then GI that is gastrointestinal tract arteriasis is there, leukemia as well as a thyroid disease. So they are infected with all these diseases. This is with reference to the Down syndrome. Okay. Then you have Patau syndrome, that is trisomy 13. Now Patau trisomy, again this is with reference to chromosome 13. There we have seen now Down syndrome is with reference to trisomy 21. Okay. Now here it is the Patau syndrome, which is actually also called as trisomy 13. Now in this case, in this trisomy 13, they have a cleft lip and palate, extra fingers and toes. They have got extra fingers and toes. Okay, that is polydactylism. If you observe their fingers and if you observe their, I mean, hands as well as their feet, they'll have extra fingers and they'll have the extra toes. And defects are actually they have infection in the heart, brain, kidneys, and mostly they're very short, and the lifespan actually is less than 1 month. Less than 1 month means they don't live for a longer duration with the Patau syndrome, trisomy. Whereas in case of that of a Down syndrome, actually it is not that lethal, but here it is lethal. This is the Patau syndrome. See here. Okay. The next is Edwards syndrome. Edwards syndrome is related with trisomy 18. So here, trisomy 18 means you have extra, means 18th chromosome. So three chromosome sets of, three extra set of what is that chromosome 18. Now here also the defects are heart defects. They have liver will be displaced, and they have the low set ears will be there. Their ears will be very low. They have abnormal hands, and severe retardation will be there, and 90% abort. They are very, very small, and their lifespan again is less than 1 year. So they cannot live for longer. duration. See, this is actually the what do you say, the karyotype of this particular syndrome. See, just now we have seen no 18th one. Okay, see here. If you see here, see this is the 18th one. So you have three sets of trisomy. It is called as trisomy. Otherwise, you have only two chromosomes. Then you have Turner syndrome. Now, Turner syndrome actually also called as exo syndrome because the people with this Turner syndrome have only one X chromosome, that is 45X. They have got only one X chromosome. Otherwise, they have two X chromosomes. The people but with this actually have got only one X chromosome. They are also called as XO. Instead of two X, they will have only one X. So that is why it is also called XO. So no Y means Turner people are female. So that means when they don't have Y means these Turner people mostly actually Turner syndrome occurs in the female but not in the male. However, no ovaries will develop, so they don't undergo the body changes of puberty and they are sterile. So that is why they are sterile. So because no Y is there and since they are female, but further there will not be any what do you say development of the ovary. So they do not undergo any body changes. They will not undergo maturity. So that is the reason why they are sterile. They remain sterile. And hormone treatment cures them, but they'll be sterile only. The other symptoms include they have got very short stature, they have got a webbed skin and low hairline at the neck, some oddities of the spatial perception, and they are not retarded. Now, hypogonadism with female phenotype will be there. They have a short body, webbing of neck, heart anomalies, infertility, amenorrhea, pigmented navy, and peripheral lymphedema at birth. So these are some of the symptoms related with the Turner syndrome. Okay, and it is seen only in the female. So this is the karyotype of this. So here, are you observing here? They have got only X chromosome, only one X. This is of the female. So if it is a female, it should have two XX chromosomes, but instead it has got only X chromosome. That is why it is called exo syndrome. Then next one is what? Klinefelter syndrome. Now, Klinefelter syndrome actually because of non-disjunction. Non-disjunction result in a person with two X's and a Y. So that is, it has got 47, okay, XXY. So this is called as the Klinefelter syndrome. Now the Y chromosome makes a person with Klinefelter male, so possessing the testis. So here it has got 2X and 1 Y chromosome. So 2X and 1 chromosome means actually it has Y chromosome. 2X actually are for female. Y chromosome, if it is there, means then it is with reference to male. So that is why the Y chromosome that is present in this particular cases or in this particular syndrome makes a person male possessing the testes. So they are often not diagnosed or diagnosed only accidentally, and they are mostly symptoms by helped by testosterone treatment, and they have a female body hair pattern, breast development, and they'll be sterile, and they can be some developmental delay or retardation will be there, especially for the verbal skills, and breast development will be there. So this is with regard to Klinefelter syndrome. Apart from these other sex chromosomes, variations are you have triplo X having 3X chromosomes, no Y chromosome means they are female. So with this syndrome, what happens? They are now till now they are not undiagnosed because they have no symptoms at all, and they have slight social and developmental problems are very few, especially language related problems are there, fertility problems are there, but many have. Most of them actually have got normal fertility and they are not well studied. So that is what is called triplo X, having 3X chromosomes. And females have 2X chromosomes that define sex in sex. In trisomy X, additional X chromosome brings the total number of the chromosome to 47. So other concurring minor conditions which are displayed by these are, it includes what? Congenital heart disease, splenodactyly, and epicanchial folds. Then facial disorders are also will be there. So this is the different types of what do you say, the chromosomal disorders or the chromosomal abnormalities. Okay. Now, disorders from changes in the chromosome structure. Now, till now we have seen the disorders related with the what related with the number of the chromosomes. But now we are going to see the disorders actually that occur or that are being caused due to changes in the chromosome structure. They are what? Deletion, duplication, translocation, and inversion. Already I mentioned that now. So deletion, duplication, translocation, and inversion. So even though there is no gain or loss of genetic material in translocation and inversion, the change in the location of a segment may alter the regulation of a gene. Okay. Now see what is deletion. Deletion, it is the first structural aberration which is detected by bridges. Okay. And here, in the case of deletion, a piece of the chromosome is either missed or it is deleted. Okay. You can either remove one or more genes from the chromosome. It occurs mostly during the meiosis. And deletions can occur in autosomes or either in the sex chromosome. And this deletion occurs due to, okay, various reasons are there. Types of deletion is terminal deletion and intercalary. Terminal deletion means, in this case, loss of terminal segment will occur. And intercalary means, in this case, the internal part of the chromosome actually is missing. So in human, genome deletion mutation are caused granulocytic leukemia and Cry-Duchat syndrome. So because of deletion, these are the two, the infections that are seen in the human. That is the Cry-Duchat syndrome. Already I have told you it is detected. Okay, it is a deletion of 5p, that is short arm of the fifth chromosome. Then duplication. What is duplication? It is the doubling or repetition of the chromosome segment. So as a result of it, what happens? A set of genes will get doubled or it will get repeated. So, and the extra set of genes, you call it as a repeat. And duplication actually was first detected by Bridges again from his genetic studies on X chromosome of Drosophila. Get doubled. Then like this, your translocation. Translocation is two pieces of different chromosomes, they are being interchanged or they are being exchanged. That is what we call the translocation. So here a portion of one chromosome will be transferred to another chromosome. That is what we call it as a translocation. Then types of translocation you have interchromosomal translocation, intraradial and intrachromosomal and interchromosomal. In intrachromosomal, you have intraradial and extraradial. Change in the position of a segment within a chromosome is intra. And change in the chromosome from one chromosome to another means between the chromosomes, you say it is inter. Intra is again of two types. One is intraradial and extraradial. Intraradial is from one location to another in the same arm. Then extraradial is from one arm to the other. So this is the different types. Then effects of translocation, it causes many changes. Okay, good example of this is on chromosome 9 and 22, creating Philadelphia chromosome. Then Burkitt's lymphoma is another type of cancer which is caused due to this. Okay. Then last is your inversion. Inversion occurs when two breaks happen on the same chromosome. So no genetic material is lost, but rearrangement may alter the gene function. Okay. And here you have two types, paracentric and pericentric inversions are there. And here the segment is turned to 180 degree or it leads to altered gene activity. That is about the inversion. Effects of inversion are, it helps to retain the original gene, it enhances the position effect, it causes the chromosomal polymorphism, and it also produces balanced gene complexity. Okay. So these are the different types of chromosomal disorders or the genetic disorders you say or chromosomal aberrations with reference to the number as well as the structure of the chromosome. Yes? Yes, students, is it okay? Hello? Hello?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Okay. I think only you are hearing, nobody is there, right?
Maybe is there one student? Ah, no, I am only there, ma'am.
Okay, okay. Okay. Whatever it is, now the time is up. Now we will close it. Okay?
Okay, ma'am. Bye. Thank you, ma'am.
Okay.